Jill, I'm
Yeah,
gonna
sure.
1, 2, 3. It started.
All right. Hello, everyone. If you're here, thanks for your patience in getting started. We had a few issues with Dr. Wegersen getting permission to share. So I will be advancing her slides from here. So don't blame her if there's any delay. So I'm really excited to have Dr. Wegersen come and talk to us today in our first virtual seminar series. She has degrees from UC Davis, UC San Diego, and San Diego State in computational biology and information systems before she moved to UConn and became assistant professor of ecology and evolutionary biology. She runs the Plant Computational Genomics Lab, and she's also director of the Computational Biology Core. Her research not only has contributed or led genome sequencing efforts from a lot of different forests and pine trees, which are massive and unwieldy genomes, but she's also helped establish a lot of new infrastructure to help the rest of the community, which is what she'll be talking about today. So without further ado, let's welcome Dr. Jill Wegersen. Thank you.
Thank you, Lindsay. I really appreciate the invitation and the opportunity to speak with everyone. I wish I could be there, of course, in person, but we all know that's the times that we're in. Given the delay, I'm going to try to keep on time and maybe go through a few spots a little faster, but I'll try to cover everything that I wanted to talk about with you guys today. I'm going to be starting by talking about kind of broadly in genomics and a lot of what we do. That's the omics we hear about a lot, but I'm also going to talk about how we integrate a lot of the other omics that are out there right now in order to achieve goals related to forest health and productivity, which is something that we think a lot about in our lab. And with that, let's go to the next slide. So I always like to start out by saying a little bit about big data. That's definitely the era that we're all in, whether we want to be or not. And a lot of the time our goal is to essentially now try to derive value from increasingly large data sets. And a few in the era or in the field of big data or information systems, we kind of talk about the 4 V's of big data. Velocity, of course, being just the tremendous speed at which we're collecting data. Volume being the tremendous amount of data that we're seeing. We're now also seeing a tremendous variety of information coming in through the same stream of information and also differing sources, and also veracity. So that reflects a lot on how accurate this data is as it's coming in to us, and us having to actually make some decisions about that as well. And I'll talk a bit about that as we move through big data. Next slide. So this is just a kind of general slide thinking about all of the omics that are out there. Even if you haven't heard it, there's probably an omics for just about everything. Like I said, we talk a lot about genomics and sort of the related aspects of transcriptomics, proteomics, metabolomics, increasingly epigenomics as well, and plants as well. But we're also thinking a lot about all of the other omics that are coming in through high throughput technologies. So related to things like phenomics, so high throughput screening, LiDAR, things like that. And so I'm going to be talking a bit about all of these aspects and also how to bring them together. Next slide. So one of the big areas, especially in forest health and forestry in general, is an increased use of technology such as LiDAR in order to monitor our forest plots. Things that we can look at such as canopy height, cover, density of trees, stem volume, age, characterizations of the species that are in that area, as well as potentially disease status as well. Next slide. And another area that we're also starting to collect a lot of information on the phenotypic side of our tree species relates to a lot of our monitoring plots. And so in specific, what was originally a lot thought of as sort of the FluxNet network or Ameriflux network, a lot of this has been incorporated more recently into the NEON inventory, so a large-scale NSF funded project where they're able to track a lot of different data types across very specific sites. And so things like phenology is just something that we're able to obtain and characterize through these plots, as well as other flex stations that might be monitoring different air quality aspects and soil as well. Next slide. And so another aspect of big data that's coming up more and more is also big data as it relates to sort of citizen science. Citizen science, of course, has the benefit of having a lot of eyes on the ground when maybe a particular researcher or scientist can only be in one place or one study site at a time. But of course, it has a few disadvantages also related to that veracity component, right? So sort of novice users trying to identify species or phenotype something in particular, or identify a disease out in the field may or may not be as accurate as somebody with expertise in that area. But at the same time, we're starting to gain a lot of information through the use of tools like that, and we're finding ways to also computationally refine that information as it's coming into our systems as well. So next slide. So I'm going to kind of start on the genomic side, and I'm going to start talking about big data as it relates to especially big trees. And so I'm going to start by talking a bit about conifer genomes. And of course, coming from the West Coast originally, some of these species, 2 of them out of the 3 will be more focused on that side, but I'll also talk a little bit about some species I've been working on this side as well. But I'm going to talk about conifer genomes because they're particularly large. And I'm going to talk a little bit about the process of obtaining a complete genome, especially in a very challenging and large organism. Talk a little bit about some of the conifer genome biology, and a little bit about what to do with the genome once we have it. And then from there, we're going to go into this idea of integrating beyond genomics and a little bit on the cyber infrastructure that we're developing in our group to enable this integration. So next slide. So the PINE-RefSeq project was a USDA-funded initiative several years long, and our goal was originally, the original call was just to sequence 1 species, and that's one that's of commercial interest, loblolly pine in the southeastern United States. But we decided that we could try to do maybe a couple more at the same time. So we also added onto that project sequencing and assembling the sugar pine genome, which is native to the western United States and parts of Canada as well. And then also sequencing and assembling Douglas fir, which also has some commercial value in the Northwest region of the US. And so our project goals were to develop community resources for these trees for the first time, obtain fundamental knowledge about conifer biology, develop new sequencing approaches, and also develop new informatic tools. Next slide. So just to put this in perspective a bit about how large these genomes actually are, you can kind of see some comparisons here to other things that have been sequenced recently. So there you have a viral genome, you have a walnut tree genome, which we've been involved in some sequencing of the Juglundaceae as well. A lot of our angiosperm tree species are less than a gig in size, so that's not an uncommon range, somewhere between, say, 400 megs and maybe about 900, so usually less than a gig. The human genome is at 3 gigs, which is common across a lot of mammalian species. And then as we start to move into some of these conifer genomes, the giant sequoia one that we recently completed is actually on the small side. It's only 8 gigs in size, but the coast redwood, which is one of the rare examples of a hexaploid species, is about 27 gigs. And actually most of the other conifers are diploid, but most of them are actually in the range of about 20 to 30 gigabases as well. And so that's a tremendously large genome. And when we were trying these genomes for the first time, they were by far the largest things that had ever been sequenced. Not too long ago, the salamander genome just came out. And so that one is about 28 gigs, and that's a really big genome as well, and had some similar challenges that we dealt with in working with these genomes at that time. Next slide. And so, as I mentioned, most of the conifer genomes are actually diploid, which is different than a lot of the angiosperm species and especially crop species that we're interested in sequencing. And in that way, it makes it a little easier. There's other aspects of conifer biology that make that process easier as well. However, they are highly repetitive, which is common across many plant species. And so you see in this diagram, they're in blue. You see a lot of our vertebrates, which are much lower in terms of repetitive content. And then when we look at our plants, things get a little bit more complicated. And those repeats definitely pose some challenges for the assembly process. Next slide. So when we started this endeavor, we were working, as I mentioned, with 3 conifer species, loblolly pine, sugar pine, and Douglas fir, but there were also 2 other spruces that were happening at the same time. So a Canadian project was tackling white spruce, and the Swedish project was tackling Norway spruce at the same time. So we were sort of all working together to see how we could achieve contiguous, more accurate genome sequences. How are we going to sequence this? How are we going to put it together? Next slide. So I mentioned that we had one advantage in addition to the fact that these species are diploid, and that's the fact that they have a haploid megagametophyte available to us, and that's huge. And so that allows us to sample the haploid aspect of the genome, which is something that a lot of groups have to try to do informatically, and I'll talk a little bit about that process as well. Next slide. And so our original strategy in doing this at the time we started these projects was primarily based on short read sequencing. And so I'll kind of be referring to the short read sequencing as Illumina sequencing and the strategies around that. Short reads, when we talk about that, are typically somewhere ranging between, say, 50 base pairs and 150 base pairs. And when we're talking about sequencing a genome, they're usually in a paired format, which means there's some sort of distance in between those 2 reads or across those 2 reads, depending on how you interpret the insert size. And so the goal at that time was to sequence incredibly deeply from as much material as possible from the megagametophyte, and then to use multiple insert sizes, which helps with bringing together contigs into larger scaffolds through the assembly process. So we were able to obtain these short read libraries, several different insert sizes, very deeply sequenced with the megagametophyte tissue. And then once we've used all the tissue that was available there from a single meg, then we went through and used needle tissue in order to generate what we would call longer insert libraries or mate pair libraries to bring together those contigs into larger pieces. Next slide. And so this is just a schematic here of roughly what the genome process looked like, genome assembly process looks like. And this is largely still the same process that is used when you think about most genome projects, and certainly things before long reads have become more prevalent, and I'll be talking a bit about that as well. And so here we take basically our DNA, we break it up into lots of little pieces, which is part of the problem and part of the challenge of the entire process. We had varying types of insert sizes. The goal is to use the incredible depth of that sequencing in order to create contigs, and from there join contigs together into what we call scaffolds. Now the final goal is generally to take those scaffolds and then join those into chromosomes, and that's still been a tremendous challenge for some of the largest genomes that are out there, and I'll talk a little bit about that. But the process of moving contigs into scaffolds can often be facilitated by these longer insert libraries or phosphonidide tags or back libraries or a lot of different technologies that are out there to basically take moderate pieces and bring them into even larger pieces. Next slide. So one of the big challenges that we ran into and kind of where our team stepped in was related to the genome assembly component. And so we actually developed a new assembler in order to tackle the problem of this tremendous amount of sequencing. At the time, generating that much short read data for a genome of that size was intractable. It wasn't something that could be really assembled on most of the computers at that time. There was one assembler that could run it, but because of the tremendous repetitive content, a lot of the assemblies were highly accurate as a result of that. And so essentially the Masurca assembler was an approach developed in a team with Johns Hopkins and University of Maryland in order to move all of the short read data into what we called intermediate size pieces, and then actually use more of a Sanger-based assembly approach, so going way back to our first generation sequencers, in order to get a more accurate assembly. So taking slightly longer pieces, then using what we call kind of a overlap layout consensus method, or basically try every piece in the puzzle until you find the one that fits. So that takes quite some time, but it ended up being tremendously beneficial in terms of generating a more accurate assembly. That being said, whenever you're developing something with short reads alone, especially something that's highly repetitive, you're still going to get a lot of pieces out of that, but that was a huge improvement. And Masurca continues to be a preferred assembler today by a lot of groups working with large genomes and also looking at hybrid assemblies between long and short reads. And that's perfect. And so one of the Masurca was eventually benchmarked across lots of different other assemblers as well, and continues to perform well for things that are highly repetitive, but almost every genome. One of the downsides of Masurca right now is the fact that it is the runtime of Masurca is very long. So on several CPU, let's see, I think we ran on about 2 terabytes of RAM and about I think 36 CPU. The job still took 2 months to run and assemble a conifer genome. So it's still a tremendously time-consuming task using sort of this modified methodology. Next slide. So after all this work, after several months of running genome assemblies, after lots of work in trying to refine that methodology, both on the sequencing strategy side and on the genome assembly side, we learned that it is indeed true. Those genomes are exceptionally large. The estimates were fairly accurate, but that short read sequencing remains fairly insufficient. We still end up with very fragmented genomes despite working very hard and going back to algorithms that are more accurate. The introns are incredibly long, and this is something that was predicted and also observed even in more common than we had originally thought. The repetitive content is very high, and so our initial estimates and published estimates are over 80%, but with better methods of detecting and characterizing repetitive content, we know that it's actually over 90%, and that pseudogenes are incredibly prevalent in these genomes as well, which gives us some new challenges related to annotating them. Next slide. So here's a view of essentially how contiguous these conifer genomes are at the point of coming out of these assembly techniques. So on the very far right column related to sequence type, we have listed kind of what the primary inputs were. Illumina in this case always referring to short reads. And then in some cases we were able to obtain either a low coverage of another technology such as 454, which is now obsolete but a little bit longer, or PacBio, which actually is one of the currently used long read technologies, but we were only able to have about 3x coverage for Loblapine at that time. And so what you see there is, though we're close to our target genome size or over our target genome size in a couple places, the fragmentation is still high. So you can kind of view that fragmentation through the total number of scaffolds that are over the length of 1 kb, so 1 million scaffolds, or several thousand scaffolds in many places. The sugar pine genome that's listed there, Pinus lambertiana, has the most contiguous genome, even though it's one of the largest, but it's also not fully complete in terms of content. And so the 10x technology that you see listed there actually takes incredibly long inserts to pull things together. It was very beneficial to the assembly, but you can still see that we still have over 16,000 scaffolds, but our N50, and that metric basically means that 50% of the genome is represented by about scaffolds of that length or longer, is close to 2.9 megabases. So a huge improvement over short read assemblies alone, but still a long ways from a chromosome scale assembly, where we're hoping to get either, most of these genomes have about 12 chromosomes, the Douglas fir has 13, but we're a little ways from that in this stage. All right, so you might say, how does that compare to other genomes that are being published for forest trees that are not conifers? And so you might take a look here and see a couple. So oak genome, eucalyptus genome that were published essentially using a lot of the same approaches, mix of Illumina and or other cases, an older one, a mix of Sanger, but with these sort of high density linkage maps, iterative assemblies, they are able to move something like 5,000 scaffolds into roughly 11 chromosomes, or 871 scaffolds into 12 chromosomes. But this advance of being able to move scaffolds into chromosomes is also still relatively recent as well. And even though these genomes are much, much smaller and much more tractable and can work on a wide number of genome assemblers, they have their own challenges as well. Next slide. So one of the challenges with a lot of our angiosperm trees is specifically related to heterozygosity. And so before we tackle some of these species, we usually go through and we try to actually estimate what that heterozygosity looks like. We usually do this with our short reads. So we take those Illumina short reads, we put together what we call a Kamer plot, and we estimate kind of what our ratio of heterozygous versus homozygous Kamers are. We are also able to figure out if we have relatively recent repeat bursts that we're going to have to deal with, or things that are more diverged. And that serves as a really nice metric to kind of decide how we want to approach that genome assembler or genome sequencing and assembly. And so there on the right is just some of the different approaches that are starting to come out. And this in particular is looking at an assembler called Falcon, where it's actually able to use longer reads in order to resolve some of that heterozygosity. So things like highly repetitive genomes and highly heterozygous genomes, both are starting to, hit a wall when it comes to relying on short read data alone. You can keep sequencing that short read data, but really short reads cannot resolve some of these huge issues. Next slide. So here's an example of a genome that we're working on right now. We've just completed the sugar maple genome. And so this is a genome that's much more reasonable than a lot of the conifers that we're working with. So it's about 600 megs in size, and through just using long read technology, so in this case the PacBio platform, pretty deep sequencing, so over 80x coverage. Despite it being a very heterozygous genome, we were able to resolve that into the 13 chromosomes that are represented there. And so it is becoming more and more possible to do this. Now, I should say that the PacBio long read sequencing alone could not produce it, though it does much better. You're in the order of maybe a couple thousand scaffolds going in, but then additional downstream processes are still needed to move it into the chromosome scale. In this case, we use the Hi-C technology to do this, which is becoming a very common methodology to approach that. All right, next slide. So just to kind of give some perspective on what plant genomes are starting to look at today, if you go over into NCBI, you'll see that there's about 477 land plants in GenBank right now. But among those, when you look at assembly completeness, about 41 are in what we call contig scale, 329 still assembled at scaffold level, and only 107 are at chromosome or what we call pseudochromosome level, which means we think we have the major pieces of the chromosomes assembled. That doesn't mean that there's nothing left over. There's usually some pieces there, but it's still a challenge. And so plant genomes are hard. And so we still see that there's a lot of work to do in this area. And most of the genomes that are assembled in that group are relatively small, with the wheat being one of the largest ones on the angioside, which is has lots of ploidy challenges going on as well. Here's another view of kind of genome sequencing technologies. There you can see the Belser et al. Put together sort of a view of contiguity of genomes as well as estimated genome size. So you can kind of see on the left side that the Illumina technologies of short reads produce lower contig N50s, but as you head towards a higher, more contiguous genomes, that you're seeing more of those long reads come into play, and that's shown in the blue and the green. And you also see some of our older genomes, which were assembled still with Sanger. Very expensive, but still highly accurate and low throughput technology, but it still produces a nice long read. And so those can still be helpful for a reasonable sized genome. Next slide. And next slide again. There we go. And so as sequencing technologies have advanced, there's been a lot of sort of rapid amount of technologies coming on the market, departing quickly. And so even though we had a lot at one point, right now we're kind of down to primarily our original Sanger, our Illumina short reads, our PacBio long read, or our Oxford nanopore long read technologies. And so most genomes are still assembled with sort of a combination of Illumina and long read, and increasingly just long read technologies alone. Next slide. And so what is the recipe for success for completing a genome? So it's not the same for everything. And that's why I mentioned that we often want to look at sort of this Kamer size estimate of the what's going on with the genome. And so that almost always requires that we do at least a little bit of short read sequencing so that we can understand, what's happening with the repetitive content, what is the estimated genome size, how heterozygous is it, all of those things. We can tell a lot from that initial estimate right there. It's also a way to detect if you've captured anything else in your genome assembly, like contaminants, for example. So those are all things that we can do when we're looking at that genome. And so here I've listed sort of some of the key and current technologies that are out there. Long reads, I believe, are for the most part required now. So if you've got a more complex genome, then you probably are going to want to include long reads, either PacBio or Nanopore, and those have reads exceeding 10 kilobases in length. And often you're going to have to pair that up with another technology. So either optical mapping, where you're using restriction site labeled fluorescent probes and using that as a guide in order to order and orient your scaffolds. Chromium 10X, which I mentioned earlier, as a way of having extremely long inserts, essentially, to bring and bridge those scaffolds back together. And then what's very popular is a lot of the chromosome conformation mapping techniques, Hi-C, that is still a short read mapping technique, which means you don't need that very high molecular weight DNA going in, but it's going to bring together those scaffolds in a ideal fashion for the most part. You're usually able to get to pseudochromosome level if you have a decent assembly going in. But all of these techniques can also be paired with a high-density genetic map, which is still one of the best tools to order and orient scaffolds, as long as you have enough density on that map relative to the fragmentation in the genome itself. Next slide. And so when we talk about developing a recipe for genomes, our group is one of a few that are part of this project, the Earth Biogenome Project. And the goal of this project is incredibly ambitious. The goal is to try to estimate all the eukaryotic diversity, sequence it in the next 10 years. It's unlikely to happen, even with our best technologies, but we're going to give it our best shot. And so one of the things that we're doing as part of this project is putting together basically guidelines on how to put together different types of genomes, handle different challenges, deal with everything from all the things that hang out maybe in a moss genome and how to get that out of there and actually sequence the moss, to what to do with a giant conifer with repetitive sequence. And so there's a couple large scale plant projects that are a part of this as well, the 10KP project, which is out of BGI in China, and then also the Open Green Genomes project, which is led by JGI. And so both of these projects are also encompassed within the larger Earth Biogenome Initiative. Next slide. And so I wanted to take a moment to mention also just the importance of this long read technology. So probably a lot of you have seen or interacted with the MinION there on the right. And so that is that USB drive style sequencer that can generate these tremendously long reads. And so Oxford Nanopore essentially took that, put it into a giant array, and created a whole bunch of other instruments, things with names like Prometheon or GridION, which you see on the other side. Next slide. And the ability, or the really cool parts about the nanopore sequencing is that it's using this pore-based technology where it's unwinding at one stage and then it's reading based on interruption of a current to call the base in real time as it's moving through this sort of pseudo-membrane approach. And so next, click. And so the capacity of this technology is tremendous. So it's also very inexpensive. So if you're running that Promethean machine with all those arrayed MinIONs, you're running about 48 flow cells. The cost is pretty minimal to get about 120 gigs of data and a tremendous N50 on the reads alone before you even go into assembly. But one of the huge advantages of this platform over other long reads, such as the PacBio platform, is that you can do things like directly sequence RNA, directly sequence DNA with minimal prep, and then also do methylation. So we can actually read what portions of the genome are methylated in real time at the same time we're sequencing a genome. And that's opening up a whole lot of doors. And we have a lot of questions and plans about this aspect as well. Next slide. And so I'll skip this next slide just for sake of time. And so with the advantage of long read sequencing, we've been able to do a little bit better job with some of the more recent genomes that have come out. So we have these species here, Siberian larch, giant sequoia, coast redwood, and silver fir. And on the next slide, I'll show a little bit of how those look. And so here, what you can see is we've got a couple of examples of where these were published with Illumina data alone, and we see very similar challenges. Over 1 million scaffolds, N50s that are lower, sort of in the kilobase level range. And then for our larger genomes that we worked on with both short reads and nanopore, and potentially also high-C, you see that those N50 values go up tremendously into the megabase range, that we're doing pretty well in terms of our target genome size. And actually for the giant Sequoia genome, we were actually able to get it into pseudochromosome level in the end, which is super exciting. And so we're getting to that point where these giant genomes are still pretty hard work, but they're a lot more accessible just in a couple of years with these new technologies. Next slide. And I'll skip this one. Next slide. Okay, so what you might realize also, especially if you're kind of diving deeper into some of our species that have been sequenced for a while, things like poplar, is that sequencing that base genome is just scratching the surface. So we are now moving more and more into the era of pangenomes, and we want the ability to be able to sequence multiple individuals across natural ranges, across different populations, and really see what's going on. We know, based on our genome sequencing approaches, that SNP level variation isn't enough to characterize what's happening in our populations, that we really do want to try to understand presence and absence of genes and how that relates to adaptation. And so this is the era we're moving into, especially with long read sequencing for lots of species, not just our model species. And so this is where we're headed next. Next slide. And then we'll skip this. Next slide. So I want to focus a little bit on some of the challenges after we achieve that assembly. And one of the areas that we spend a lot of time thinking about is genome annotation. When we talk about genome annotation, we're talking about the structural identification of the genes on the genome itself. And we generally do this with a couple different approaches. We align external evidence in the form of transcriptomes, and then we also try to train machine learning tools in order to identify genes against the genome at the same time. And then we usually have a secondary step in that process that tries to provide or assign functional annotations to the genes we detect. Next slide. And so one of the challenges with our genomes was not just the fact that the assemblies were fragmented initially, but was also the ability to try to identify the correct number of genes. In a lot of cases, we were either inflating that number initially or underestimating that number. As the contiguity of the genomes improves, the process gets a little bit better, but it's still very challenging for lots of other reasons. Next slide. Okay, so just a reminder here, when we talk about challenges facing the genome, this is just another view of the size. Next. And then the fact that, of course, while we might be aiming to have about 12 chromosomes, we actually have tremendously more pieces, and that we've got genomes that are fragmented. And that we also have this tremendous repetitive content, right? Like I said, 85 to 90% of these genomes, potentially more, is going to be repetitive content. And then we mentioned that for every pseudogene that we identify, probably 5 others that look like genes are actually pseudogenes. And then we also have these uncharacteristic gene structures. So I mentioned the fact that the introns are incredibly long. So some of the recent estimates, we've actually even found longer than that. So about 1.3 megabase long introns within a single gene. And that does not play well with a lot of approaches that we have available to us today. Next slide. And so one of the things that's very important for genome annotation is to obtain high quality transcriptomic evidence. This is especially important when we have some uncharacteristic structures that we're working with. The 1KP project has been a tremendous resource for kind of a nice diverse range of plant species. Next. And so, okay, if you have good transcriptomic evidence and you have a decent genome assembly, why is this still hard? And so when we're aligning resources to the genome, we find that there's actually quite a bit of variation, especially because the transcriptomic evidence often doesn't come from the same individual that the source genome was assembled from. We know we still have errors in our genome. We know that we also have incomplete models that we're aligning with to begin with. So de novo transcriptome assembly produces and includes some errors in and of itself. And in addition to that, we do still have errors in our assembly. So we may have gaps in the assembly that are causing some challenges in that as well. When it comes, go back, sorry. When it comes to ab initio approaches, we have to train based on our alignments in order to find and predict the proper genes. So we're usually doing that with protein to genome, gene to genome, and exon level support. Next slide. And next. And so with the existing programs that are out there, next slide, we get a huge range of estimates of gene space when it comes to some of our conifer species coming out of these pipelines initially. So we're getting estimates that clearly exceed what we would expect to be in a eukaryotic genome. So almost 200,000, sometimes exceeding 200,000 in some cases. Next slide. And next. So we go through a lot initially of processes, kind of iterating through different metrics, what's happening with our exons, our splice sites, what's happening with our mono to multi-exonic ratio, all of these different things. Next slide. And so in the end, another way that we also benchmark our genomes is also to look at these, what we call BOOSCO genes or orthologs, and that stands for benchmarking single copy orthologs. And here we use about 1,400 what are believed to be conserved single copy genes across land plants. And we try to see whether or not we can find them in the genome and then in our resulting genome annotation. And then how does that compare to our transcriptome that went in? We also know there's not a lot of gymnosperms that went into consideration of developing the BOOSCO benchmark, but we know from looking at the transcriptomes, especially those that are deeply sequenced, that we should be able to get into the upper 80s in terms of percentage of conservation. And so what we see is that when we run these programs against the genome, the long introns start to cause a problem, because if we estimate across the genome, we should in all cases do the very best. But in fact, we sometimes have gene estimates that exceed that because those are run on the proteins rather than the genome itself. And so either way, when you look at our more fragmented genome, we still have tremendous issues, right, with a genome level and gene level estimates. When we look at our conifers, for example, we see the same. We're still not really getting much higher over kind of the upper 50% mark, with the exception, of course, of Needham, a gymnosperm that was recently sequenced, which is doing a bit better at 72%. Next. And so as part of our software development kind of next stage, after putting together approaches to improve upon assembly of complex genomes, we decided to tackle annotation of complex genomes. And so here, and we can kind of just push through the workflow, I think, on the slides, we developed an entire workflow that works from a combination of RNA-seq short reads as well as protein sequences that typically come from de novo assembled transcriptomes. These get aligned into a preliminary program called Breaker, and then we do a series of filters from there based on ab initio models, using a couple tools that we developed in-house, one of those being GFAX, and the other one being NTAP, which uses the functional information in order to filter the genes on the other side. So next slide. And so I just want to say a couple notes about these programs, and we can slide through the notes on those. The NTAP package is specifically designed to work on both transcriptomes, de novo assembled transcriptomes, as well as gene models. And one of the values in using this is it's able to assess the predicted gene through a variety of metrics. And by doing this, it's looking for protein domains, it's looking for any sort of functional signatures that we actually have a correct reading frame and we actually have the correct gene. And so that's something that the NTAP package can do, but it can also do that for transcriptomes as well. And it sits at the end of our annotation pipeline, and it also sits at the beginning of others' de novo transcriptome assembly pipeline as well. Next slide. And the other tool that we use in this package is the GFAX tool. And this is actually responsible for lots of different structural filters. And this is a very key component because a lot of the ab initio predictions coming out of our pipeline that give us those numbers that are over, 200,000 in some cases, need to be further filtered down. And so the GFAX package assesses on a large variety of different structural elements, things that we know about the species that we can put in, such as intron length, things like that, and try to see whether these are correct or not. And so the combination and iterative process of moving with both GFAX and NTAP has provided an improved annotation pipeline that is working on some of these more complex genomes. But in addition to that, we currently have sort of a new project underway right now that we're actually kind of redeveloping annotation from the ground up. We're using some deep learning approaches in order to do that. So we're just at the start of that. And we're actually going to be using folding information as well as functional and structural primary sequence data in order to improve the predictions of these gene models even further. Next slide. And so what we're able to do out of our pipeline is bring some of these initial predictions anywhere from, really early on, over 200 to 300,000 genes, if we don't do any filtering. Our recent estimate coming out of Giant Sequoia, even though that was a very contiguous genome, almost back to its 8 chromosomes, was over 1.4 million genes initially out of that. And so by using some of the refinements in the couple tools that we produced, we're able to get that down to a much more reasonable number. Next. And so we're kind of comparing that here, and we can go across to several other sequenced species that are out there. Conifers, gymnosperms in general are not thought to have more genes necessarily as diploid organisms than other species. So we expect to hit somewhere between the 35 and 45,000 mark for the final number of genes we would expect in those species. Next. And so here's just a kind of an example. And I'm going to skip, I think, this slide in terms of more benchmarking. But right here, I just wanted to mention that after that process, we've been able to also further look at challenges around sort of our mono and multi-exonic genes. Monoexonic genes are a real big problem when you have a high percentage of pseudogenes in your genome. And so we've been also looking at other sequencing approaches in order to improve that. And so ATAC-seq is one of those approaches that we're using in order to actually look where we have actual chromatin binding and say, yes, is this likely to be a real gene? And so we have that information there too. So next. And I'm going to skip through these next slides. Next, and next, because I want to get to this part. And I apologize, just because of the technical delay, I want to make sure you guys get kind of a high-level overview of everything. So in short, or in summary of what I was kind of just talking about, essentially we've developed and focused on developing assembly approaches to improve upon complex genomes, and then following that, annotation approaches in order to refine gene models, call them more accurately, obtain functional annotations that make sense for those refined set of genes. And after all of that, we're able to start to ask some questions. That's when we can move into things like, can we start to sequence a pangenome? Sorry, one back. Can we start to sequence a pangenome of this species? Do we have a high enough quality reference to do that? Do we really know where the genes are? And set up from there. But what I want to talk about a little bit is now moving more into population studies and this idea of integrating beyond genomics and doing this effectively. So capturing all of that other data that's out there. And I just want to start, so next slide, and talk a little bit about one of the databases that we curate. So the TreeGenes database is a database that's been around actually since the early mid-90s. It was under the name Dendrum at the time. It was an original ARS funded database just when the internet was becoming a thing. And so since then, so it's been around for a while. It's changed names, changed platforms a few times, but essentially its goal is the same. So it's there to curate information on the genomes and the phenomes and genetic maps and markers of forestry species around the world. And so today that database holds a little over 2,000 species, or almost 2,000 species, and we have several registered collaborators, and we freely distribute and disseminate information that is related to these species, both information that's curated out of public repositories, text mining type of information coming out of literature, genome sequences as you might expect, and a lot of other information for the community, research community as well. Next slide. And so this database is actually developed in a framework that we call Triple. and this is an open source framework that we've been funded to support and develop for several years. It brings together analytics, backend analytics through something called Galaxy, ties together a database backend that we call Chato, and then it has a modular front end through what we call a content management system known as Drupal. And these components work together with some default modules that do things that most genetics and genomics databases might want to do. Things like have a genome browser to show a genome, or ways to search functional annotations, or ways to blast a sequence against a database. And so these are all maintained and community developed as part of this framework. And Drupal in itself has actually over 100 instances of databases around the world today. The I5K arthropod database is held in Drupal. There's even a more recent COVID-19 database that's been sprung up and hosted in Drupal. So this is a framework that's very flexible to holding a lot of data and is also the backend for the tree gene system and kind of part of our development goals in the group. Next slide. And so part of working within Drupal is that we develop a couple modules for the system that can plug in, and anybody in the community can develop these within the Drupal framework and share them out. And two of the ones that we specifically develop for tree genes are the Plant PopGen Submit pipeline and the cartography application. So next slide. And so just to give a quick overview of Drupal Plant PopGen Submit, our goal here is to take in metadata and raw data on population and association genetics type studies in trees initially. We've now actually moved into being able to take that information for plants more broadly as well. And so this pipeline is actually intelligent. It's going to say, tell me a little bit about your study. And once it hears a little bit about that, it's going to ask you questions that are directed towards the type of study you have. And a lot of them are, is this a genotype phenotype? Is this a phenotype by environment? Do we have all three? Are your trees georeferenced? And so on. And so we'll actually walk you through some questions and then produce very directed requests for data, check things. It will take everything from the raw data itself to descriptions of the phenotypes and so on. Next slide. And so one of the goals, sorry, one back, you were good. So one of the goals of the Drupal Plant PopGen Submit is also to specifically focus on phenotypic data. And so we work with several different community curated standards. One of them is MyAPI, which is minimal information about a plant phenotyping experiment. And what we take in there is basically, can we standardize, can we map all the different things that were controlled? Is common garden? Is this in a growth chamber? You know, how is the study controlled? How are things measured? What units were used? And that information is collected and mapped onto the standards document because it'll make it a lot easier for individuals to search, query, or perform meta-analysis downstream because all this data is collected on the study. We also provide a DOI, so a long-term DOI that's associated with the data and how's long-term. And so these studies can be linked and referenced in publications once they're submitted. We get a lot of direct submissions by working directly with journals, and so journals will send people there to submit their data. We're going to be working with more journals since we were extending out to plants just from trees as part of our current USDA initiative. But right now, this has mostly been focused on trees, and so we work with those journals and have them help us, you know, direct users over and then send back to them a nicely captured study with the metadata from the start to the finish of the study, including all the analysis as well. Next. And so once you have that accession number available, that study lives on the tree genes. And so it's something that you can search by the study, by the raw data, by the phenotypes. You can dive into each of the data sets and access it there. And then you can also actually go and view it on some of our analytical platforms as well. And that's where I'll talk a little bit about the other module that we develop besides tips, and that is cartography. And so cartography is a map-based, web-based application that brings together genotype, phenotype, and environment for georeferenced plants. We've just made the transition over to more plants, so right now it's primarily still trees. So when you open up the cartography application, you're able to sort of navigate around the world. You're able to see anything anybody submitted to us, as well as a lot of different studies that get brought in different ways, and those can be curated, and those can also come through some of our citizen science applications as well. So when you come in initially, you kind of see everything that's available. Next. And then you're also able to turn it on and off different data set sources. So anything coming through TreeGenes is information that was submitted as studies through TIPS. We have TreeSnap, which brings in actual observations. So we wouldn't normally have sequencing data on those for the most part, though researchers are increasingly using TreeSnap as actually a phenotyping app themselves. But you would see, for example, phenotypes that might be called for, and we would have georeferenced information on those trees. The way TreeSnap is designed right now, it actually allows the researcher to specify specifically what type of phenotypes they want to capture on species. So if they're interested in targeting information in a region or in a specific species, they can actually put that into the app and get some citizen science assistance in doing that. We also curate data out from external sources. So we have Dryad on there as well. And you'll see when you expand the environmental layers, this is one of the key benefits or features of cartography, is we have several different environmental layers, and we're starting to also pull in different LiDAR layers as well and learning how to do that. And so with these layers, you can turn them on and off. You can change the transparency, and you can also select any point on the map and obtain data at that location. And all the data for where we have a plant that's georeferenced in the database, we can pull all of the metrics from all of the layers from the database in real time with whether or not those layers are loaded or not. And so you can go in, you can point on any portion of the map and get environmental information. You can also click on a tree and you can understand what study that came from, how it was linked, source information, get more images depending on if there are images available, phenotypes, and so on. And you can actually start to collect either entire studies or individual trees into an analysis. And down below, which I'm not showing all the filters, you can actually search by different traits, genotypes, marker types, an entire study, and so on. So as you kind of scroll through, there's many different ways of interacting with cartography and pulling together different studies for meta-analysis as well. Next. This is just an example of some of the different layers that we have on the map. So things that allow you to look at range maps, you can look at WorldClim data, soil, canopy, and so on. And so here's a view here of looking at an individual tree. Just as you might expect, some studies are going to be looking at the individual, have individual coordinates for each tree. Some will be more site-based. And so we have different ways to represent common garden style studies or to represent more like general site applied to multiple trees. And so you have ways to kind of evaluate how accurate those coordinates are when you're performing your analysis as well. And so that's information that is included here as well. And here we have the tree cover, one of the tree cover layers that is loaded. Next. And then once you have all of your particular individuals loaded, you've worked through the map, you've searched by study or type. Next, you can then start to obtain more of a list type view. You can start to actually visualize some of the trends in the data and the type of trees you've selected. Next. And then you can actually move into analysis. And so one of the aspects of a triple framework that I mentioned early on, we've got a web front end, we have a database back end, and then we also have an analytic engine, and that is provided through this tool called Galaxy, which is actually a widely used platform for doing informatic analysis. On the back end, we are actually developing workflows that are intelligent. So those workflows say, oh, you've got this type of data. These are SSRs, or these are SNPs, or these are whatever you have, and this is what's actually available to you. This is what you actually can do with your data. And then this is the kind of filtering you might want to do, and so on. So we try to make those workflows sort of as intelligent as possible, especially for users performing meta-analysis across studies that were submitted through the TIPS pipeline, where we know enough about that study in order to say what is actually doable. Next slide. And so when we're building workflows in Galaxy, we can do this sort of all command line, but we can also do it visually. So you can take a lot of our common open source bioinformatic tools, we can put them together, we can add constraints, preset parameters, and we're doing a lot of this in order to develop these backend pipelines that can be used with all those TIPS, those studies that came into TIPS initially. Next. And so at a very, very basic level, a lot of the workflows we've been focused on are related to landscape genomics, which makes a lot of sense for our forest trees. And so at the base level, we're trying to bring together genotypic and environmental data. We're trying to crunch all of that in Galaxy and get some correlated markers. Next slide. And so here, this is actually in detail what it actually might start to look like. The workflows are much more complex. We have to do things like pre-process our data, filter by individuals, clean up some of that SNP data, remap onto maybe a new version of the genome, which we have the ability to do as well with enough metadata, estimate population structure, look at things like heterozygosity, and then hopefully come out on the other end with some results. And so there's a lot of ways and sub-workflows that work with the user in order to walk you through that pipeline. And so in cartography, that's kind of been our focus. We're continuing to develop these workflows for our end users. We're working with kind of our advisor groups to see what people want to do. And we've been working a lot with the community in order to get people to send us some data. So if you have studies like this, we welcome receiving them. We have a curation team that actually goes through things like Dryad and pulls things out of publications. But for any of you researchers, you know how hard it can be to find all the information you need in those supplemental files. And so this really provides a way to make this data accessible to the community and also allows us to build on a lot of that research that's already taken place. Next. And this is just sort of a conclusion slide and the value of landscape genomics in order to provide tools in order to basically predict what's going to happen, maintaining adapted and healthy forests as we move into our changing world. And with that, next, I just want to acknowledge lots of wonderful people that we work with. We are highly collaborative because we focus in the areas of software development, tool development, genome assembly, and things like that. So we're always working with lots of different groups and ready to take on new and challenging problems within plant genomics. And next slide. Ooh.
Sorry, I closed something.
No, don't worry. It was just a picture of our team. You're good. So that was the end. So thank you guys. And I apologize for rushing through a few components. There it is. Yeah, so that was our just our team that's done a lot of the hard work on cartography, Masurca, as well as our packages, easel and GFAX as well. So thank you so much for hanging out through the slightly rushed presentation. And I'm happy to take any questions if we have time.
So anyone can put a question in the Q&A. Oh, Wade Elmer says, are the cartography tree data sets available to everyone, or are there fees for users?
No fees, everything is free. This is currently a USDA funded project, and so the data sets are freely available. And we do allow users to sort of upload their data if they're in the process of publication, and we will hold that data just like GenBank does until publication. So we do have some data that's just going to be behind the firewall until the paper's out, but everything else, as long as you okay it, that data goes out. And so most of our data currently is completely publicly available.
Well, thank you so much. That was really exciting talk, and I would love to explore some of those resources you've built and look forward to what you're building in the future with your new NSF career grant and all the other projects you have going. So I know a few.
Sorry, Lindsay, for interrupt. There's another question from Kwan. He asking about what is the evolutional physiological significance for some of the conifers to maintain such large size repetitive genome?
Right. We don't necessarily know all the answers. Something that we were looking at in specific was, and what I actually kind of skipped through with a bunch of slides, was related to the long introns. And so something that we were actually focused on was like, how does the entire RNA machinery even handle these very long introns, and it's a very energy exhausting process. And so we actually identified some regions in the transcription factor 2B protein that had this very conserved and very large insert across all of the conifers that have these long introns. And it's a very disordered and flexible region in the protein. And so I thought that was kind of interesting. It's all informatic. We haven't been able to, validate that in another context. But it definitely plays on the idea that there are some different mechanisms at work, especially for long introns, of which where we see a lot of our repetitive content nested within our long introns. And so in addition to that, we've also identified the fact that there's a lot of the cleanup mechanisms, specifically some of the dicer proteins that are involved, are not present in some of our conifer species. Like they're present or expressed in embryo, but they are not expressed beyond that, which means that, you know, as these trees mature, the ability to actually clean up some of this extra repetitive content is lost. And so we've seen that as well in some of those species, but exactly why that's happening, still some unknowns there.
And I have a question. How much of the gene content or the annotated gene content has totally unknown function compared to, you know, more model plants like rice and Arabidopsis?
Right, a lot. So for the conifers, I would say that about 65% or so, we might be able to say, you know, we have a nice Arabidopsis ortholog. We know something about what that gene is likely to do, but outside of that, there's a lot of, you know, unanswered questions. And I think that's been a big area of interest, is sort of, we don't really have any sort of representative model within the genomes at all, and getting towards that so that we would actually be able to uncover that. And there's some programs I know that are dedicated to taking some, right, basically creating more models. Creating a model of gymnosperm is tough. They're not as amenable, right, to a lot of manipulation as other things are, but some are a little easier than others. But we would need to get to that place in order to say more. But right now, I'd say about 65% is a good estimate in terms of functional knowledge.
Well, thank you so much again. I'm not seeing any other questions, so if no one has more questions, I'm sure we're all giving you a round of applause in our separate offices. And I hope we get to meet you someday when we can all get to know each other again.
Sounds good. It was a pleasure. And feel free to email me, of course, if you have any questions or thoughts or want to use any of these programs. We're happy and willing to help.
Okay, thanks very much.